focus of our lab is pollinator health. And I realize almost everyone here is doing plant-related things, and this probably sounds very animal-related, but what I'm going to show you is two of the things. So we're really focused on the interface between the plant and the insect. So like a lot of labs at BTI that also focus on that interface, ours just happens to be pollinators. So not necessarily the things that are eating the plants that we might not want them to eat, but the things that are actually benefiting those plants. We have a fairly large lab, so I just want to introduce you to at least an overview of what the lab is. And now Bliss is in the lab for this summer working on one of the projects that I'll tell you a little bit about. And also Elena, if she's here. There she is. Great. So Elena is here working on the other project that I'm going to tell you about. So I focused in on two of the projects, mostly just to show you guys a little bit some background on what these projects are, but also give you guys an overview of what we're doing. We have a pretty big lab. We're in the Department of Entomology, and then the people who are not in the snowy photo here, but on the other end of things, we actually run an extension program too. So part of what my role is not just doing research, but also extending that information to the public. So instead of a lot of people at Cornell who have a teaching and research split, I have a research and extension split. So even this morning I was actually interacting with a whole bunch of beekeepers, which is probably why I look a little haggard from being up very early this morning. Okay, so just to give you guys a little bit of introduction to pollinators and why we care about them, let's start with, who are these pollinators? Do you guys know how many species of bees? If you were to just take a wild guess, how many species of bees do you think we have in New York?
I thought it was over like 900 or something like that.
900 species, okay. Anyone else have a guess? 300? 3? You guys are in the right ballpark. I'm really impressed. So usually people say something in the range of 30 or 40 or something. Whoops, wrong way. 416 species. So really good. I'm impressed. Have you guys seen one of my talks before? Is that why? So we have 416 species. We have 3 main managed pollinators here. I apologize. I don't know why this is a little grainy. But 3 main managed pollinators. So by far our most abundant is on the left-hand side here, the European honeybee. So Apis mellifera. Is anyone a beekeeper in this room? No one's a beekeeper? One beekeeper? Okay, so we have one person, 2 bees. That's right, actually we have bees on your property now, as of this morning. It's actually somewhat ironic, I didn't realize that until just now. So the European honeybee is our most abundant. We also have Megachile rotundata, the alfalfa leafcutter bee, and Osmia cornifrons, a mason bee, that are our main managed pollinators. We have one that straddles the line. So this is the common eastern bumblebee, Bombus impatiens. So it's used for pollinating primarily tomatoes. Somewhere around 80 to 90% of the tomatoes that you eat are pollinated in greenhouses by that particular bee right there. A whole industry has sprouted up around managing that bee. But it's also our most common wild pollinator that's here in New York. So it straddles the line between both managed and wild. And then we have 412 species of bees that we don't know a lot about. So here are the breakdown of all of these different bees. We have 168 species of solitary ground nesters, 58 species of social ground nesters, 30 species of social cavity nesters. We have solitary cavity nesters, we have social cavity nesters, kleptoparasites, so parasites of bees that are also bees themselves. We have stem nesters, wood nesters, resin bees, and even a snail shell nester. One of the main questions I often get from people is, what is the difference between a social bee and a solitary bee? So most of you probably think of social bees when you think of bees. This is the European honeybee. We put it in boxes, and they have these massive colonies. But there's actually so we have 73 species of those, these social bees here in New York. The vast majority of them are not social, they're actually solitary. So what are these social bees? They have cooperative brood care, overlapping generations, and division of labor. We won't go into this since it isn't an introductory lecture on beekeeping. But just some pictures of this. So one social cavity nester, this is our common eastern bumblebee, Bombus impatiens. You can also stick this one in boxes. This is what's used to pollinate a lot of your tomatoes, and you can even bring it up to the governor's mansion in Albany if you want to, and show them what the bees look like. They'll let you do that. Then we also have our most famous social cavity nester with someone who is related to someone else in this room and on the left-hand panel here. But we have the European honeybee. We stick this one in boxes, and then we steal their honey at the bottom in these giant centrifuges. Okay, so we also have 343 species of solitary bees. So this is the vast majority of bee diversity that's here in New York. It's also the vast majority of bee diversity that's in the world, okay? So what do these bees do? Something fundamentally different. So they have single parent care. They do not have these massive colonies, but they have a single individual parent who is now laying eggs and making little individual pollen provisions for each of those eggs. And they have this discrete generation. So typically one generation per year. It's not this continual overlapping generations that's making this massive colony of bees. And they have no division of labor. So they're just all doing their own thing. Okay? So we won't get, happy to talk more in detail about bee biology if you want to, but we'll get a little bit further on in the intro here. Okay, so just some nice pictures. So here's a couple solitary ground nesters. Here's, I think, maybe the cutest picture of a bee that I can possibly find. This is the wool carder bee that actually shaves the trichomes off of lamb's ear and uses it to line its nest and make just a beautiful place for its offspring. Solitary wood nester, anyone know what this one is? Carpenter bee, that's right. So some bees are excellent pollinators, but also considered pests if you happen to have old wood, around your house. Okay, so what are these bees doing? They're going out, and we call it pollinators, right? But what they're actually going and doing is functioning as an herbivore. Okay, so they are going out and collecting nectar for their source of carbohydrates, and then they're also collecting pollen for their either protein, vitamins, or fat. Okay, so they're basically going out and trying to make their living off of flowers. Here's just some pictures of different bees doing the exact same thing. But while they're doing that, they're also transferring the male pollen to the female ovary. So they're transferring gametes to facilitate plant reproduction. Okay? That's really good for natural systems, but it's also incredibly important for our agricultural systems. So here are all the pollination-dependent crops in New York. Apple is a big one, so we're called the Big Apple State for a reason, worth about $240 million annually. We also do soy, beans, squash, cherries, lots of these high-value fruits and vegetables that rely on pollinators. If you total all of those up, depending on the year, it's worth a little over $1 billion in terms of how the gross value of all of these pollination-dependent crops. If we then consider the reliance on pollinators, so apples, for example, 100% reliant on pollinators. You're not going to have an apple fruit produced unless a pollinator is there. Soy, on the other hand, can do actually quite well without pollinators, so only about 5 or 10% reliant on pollinators. So if we total all of this up, we estimate that it's somewhere around $500 million annually that pollinators are contributing to our agricultural economy. Okay? That's just in New York State. Worldwide, it's worth about $170 billion per year. And then I'm sure you guys have heard this statistic before, but somewhere around 3/4 of the world's crops rely on pollinators. Okay? So pollination, very important for natural systems, so the functioning of ecosystems out there, but also very important for agriculture. Okay, in addition to that, honey is worth something, and beekeepers are worth something. So we have about 3,000 New York State beekeepers. They keep somewhere around 100,000 colonies, and they produce about 4 million pounds of honey annually. So that's worth, we think, somewhere around $10 million per year. However, this is what's going on for the last 3 decades. So if you look at New York State honey production in millions of pounds here, starting in 1987 and going all the way up until I think either 2016 or 2017 here, we're seeing a drop in honey production over this time. And you guys have probably heard about pollinator declines in general. This is one potential thing that's going on here. So in fact, the last, I think this is 13 years of the last 3 decades. The most recent 13 years were actually the lowest honey producing years in New York State's, at least recorded history. Why is this occurring? It's because of, well, part of the reason is because of these high loss rates that we're experiencing for honeybee colonies. So this is just a map of the United States showing estimates from 2016 to 2017 of percentage losses of bees, of honeybee colonies in those areas. So New York here, you can see, we're losing somewhere around 44% last year. It's averaged somewhere around 50% loss rates for the last about a decade or so. So of those 100,000 colonies of honeybees that we have here in the state, we're typically losing about 50,000 of those every single year, okay? Massive losses that are occurring. At the same time, so if we go back to these 416 species of bees in the state, we know that 53 of those 416 species are experiencing range contractions or population declines. Okay, so those bees are becoming either less abundant or where they used to actually live is being shrunk into a smaller geographic area. Okay? So these declines, this is what a decline is, this is what you see in the news, this is what those declines actually mean. Okay, and then the last point I want to make is, this is a data set that's coming out, or has come out of Europe just this past year, looking at total flying insect biomass starting in 1989, and then looking over for 27 years how insect biomass in natural areas in Europe has been essentially declining for the last 27 years. I think likely we are going to start seeing more of these data sets. You're going to see more of these in the news. This was highlighted on New York Times, CNN, other popular media. I don't think it's just pollinators that are experiencing declines. I think it's insects that are experiencing declines. This is my opinion right now. We have one real good data set for this, a few other things in the works, but be watching for that, I would say, over the next 5 years or so as more people start to publish these data sets. Okay, why? So the question is, why are these declines occurring? And then if we actually understand why they're occurring, can we actually do something about it to ensure that we have bees around, you know, for the ecosystem services they provide, but also for the agricultural services that they're doing. So the pollination that they're doing for, I'm sure, what we're going to be eating for lunch. Okay? So what are the main reasons for why pollinators are doing so poorly? So the first is exposure to agrochemicals, so being exposed to insecticides, fungicides, and herbicides. Second thing, pests and pathogens, so especially novel pests and pathogens like the varroa mite. If we have one beekeeper in here, do you treat for varroa? Yes, okay, good. So varroa mite is one of these novel pests that made a host switch onto Apis mellifera. That has occurred in several other bee species with other pests, and those are the pests that seem to be posing the biggest challenge for bees. Climate change is also a problem, so things like big droughts that occur, for example, in California, where we do a lot of pollination of crops like almond, and then phenological mismatch, which I won't get into. Loss of habitat is potentially one of the biggest factors. So as you have agricultural, especially agricultural intensification of areas, you lose a diverse array of things that bees are able to eat and also make their homes out of. And then you have these monoculture cropping systems that pop up. Those might bloom, but they only bloom one time of the year. So it's this mass bloom, and then it's just a desert for bees for the rest of the year. Okay? And then final thing is poor management practices. So poor managed bees, especially honeybees, which I'm showing you here on this big 18-wheeler truck, if you have poor management practices, for example, you're not managing for the varroa mite, then there can be things like pathogen spillover, which is one of the topics we'll talk about in a little bit here. Okay? Okay, so there's an introduction. What I'll do is I'll focus, so we have, I think, around 30 minutes or so I'll try to spend about 15 minutes on each of these topics. One project that we have going on with looking at the exposure of pesticides, so what pesticides are bees exposed to, and is that posing big risk, and when is it posing big risk? And then I'll finish up with a project that we're working on related to pests and pathogens, looking at transmission of pathogens at flowers and how that's impacting bee health in general. Okay, so we'll start with pesticides. Any questions before I go on, by the way? You guys have your intro on bee health now? You're ready to go. All right, good. Okay, so we'll start just by showing some data that we've been taking for the last couple years as part of this thing called the New York State Beekeeper Tech Team. So this was started in 2016. We've now done this for 2.5 years. And one of the things that we've been doing with the tech team is going around and looking at pesticide exposure to all the different honeybee colonies throughout New York State. There's a lot of other stuff that we're doing, but I'm just going to focus on that for here. So here's a picture of Emma and Mary Kate going out and actually sampling some of the pollen from these colonies to then we'll take it back to the lab and look at pesticide residues. So here's just them taking some pollen off of a frame. And then this is Nico Baert, he's a chemist in our lab, and this is our LCMS, so we do liquid chromatography. Is anyone actually doing an RU or a project with chromatography? One person? Okay, so you guys know, you at least know chromatography. Does everyone know everyone else know what chromatography is? Okay, good. We can chat about it more in detail later too if you want. But anyways, this fancy machine allows us to look at pesticide levels in any environmental sample, including pollen and wax. Okay, so we went out for our first year and looked at 198 colonies throughout New York to see what types of pesticides were in those colonies. Here is our top list of our just 13 most prevalent compounds that we found. And we find a mixed bag of a lot of different things. So piperonal butoxide, a synergist, was found in almost every single colony. So this is actually a pesticide that causes insecticides to become more toxic. So oftentimes if you have a can of wasp killer, it'll have pyrethroids in there, but you'll also have piperonal butoxide to cause those pyrethroids to become more toxic. Okay, so immediately, what we can already say is if we're looking at risk, we probably are miscalculating risk because we don't know what the interactions are that are occurring with piperonal butoxide and some of these other things that are potentially causing interactions. Okay? But let's try to get to risk, and I'll show you how we get there. So lots of different things, including lots of fungicides. I guess that's the other major take-home, is that we find lots of fungicides in there. So here's a histogram showing the number of pesticides per colony for all of those 198 beekeepers that we went and sampled. So on average, we're finding about 6 compounds, so that's the median, is 6 compounds per colony. And this is broken up by commercial beekeepers, so sort of large-scale guys that are really making a business out of this. In red, sideliner beekeepers, in orange, and then just hobby beekeepers if you have a colony or 2 in your backyard. So the major take-home message here is pesticides are ubiquitous. So bees are exposed to pesticides pretty much all the time. There was one hobby beekeeper that had no pesticides in their colonies. Every single other beekeeper in New York had at least some detectable levels of pesticides. Okay? I think that's the main take-home from that. So what about risk? So for risk, what we look at is not just whether a pesticide was present, but what is the concentration of that pesticide and what is the toxicity of that pesticide. And we can do that with some math that I'm not showing you here, but we can do it on the y-axis here looking at a wax hazard quotient. So this is basically your percentage of a contact LD50. You guys know what an LD50 is? So a lethal dose of killing 50% of organisms. So your LD50 here, if you're at 100% of your LD50, you're acutely killing 50% of the organisms in that honeybee colony. what you can see here is for all of these commercial beekeepers, and the same was true for sideliners and also hobbyists, we're really low. So even though we're finding a lot of pesticides out there, they're at really low levels or they're not very toxic to bees, okay? So in general, we're at like, less than 1% of an LD50. We're not really killing all that many bees, but they're there, okay? Okay, so major take home, generally low risk from pesticides if you just go and look at people's bee colonies. All right, now let's go do some commercial apple pollination. All right, so this is now a different project. We're not just looking at where any beekeeper is having their hives and what types of pesticides they're exposed to. Now what we're going to do is we're going to go and mimic what these guys are doing down here. So most of the apples or these large fruit trees that are grown in the United States rent honeybee hives to come in, and then those pollinators, they're sort of contract services, right? So you come in and you drop your bees there, and those bees fly around to all of the different flowers and do the pollination, okay? In New York, apple is our big one, and what we did is we basically tried to mimic commercial pollination to see what kind of pesticides the bees were exposed to and how they were exposed to them in apple. So these are locations of the 30 different orchards that we went into, and then just a picture of what our colonies look like down there. There we go. Oh boy, forgot I had this in here. Here's our cartoon of how we did this. So 4 different colonies for 30 different apple orchards here. So 120 colonies total that we put out. Okay, so what did we do? So we sampled the colonies when they were in doing apple pollination, and we looked at 2 main things. One is we scooped out all the pollen, and we looked at what types of pollen they were actually bringing back. So on the right-hand side here, you can see under a microscope with some stain what apple pollen would look like. And then on the picture below, you can see what buckthorn pollen would look like, just a different type of pollen grain. So we looked at all of that across 30 different apple orchards and basically figured out what things, or what the whole distribution was of what they were bringing back. What I've highlighted here in red is the amount of apple pollen for each of the 30 orchards across the x-axis here. And then in purple was the dominant pollen, so that was buckthorn. And then all the other types of pollen are in gray there. So 2 major things, take-home messages from this. One, apple pollen, they're going in to pollinate apple, but they're actually not bringing all that much apple pollen back. So they're actually pollinating a lot of other things other than apple. And buckthorn is our major one. Do you guys know what buckthorn is? So it's an invasive species. Here's glossy buckthorn right here, sort of taking over an area, and it blooms about the same time as apple. So our contract service, providers, our honeybees that we're bringing into apple orchards are actually doing a lot of invasive species pollination, not necessarily apple pollination. Okay. Then we did the same thing, so we go back to Nico here, and we looked at what types of pesticide residues were in the pollen that we also identified all that pollen from. And what we found, so what we found is this. So this is basically just a heat map showing all of the 30 apple orchards along the y-axis here, and then the types of insecticides and fungicides and herbicide that we were looking at across the x-axis. So if it's a red, there were large amounts of that particular pesticide found. Orange, low amounts, and gray, there was nothing found. So the main take-home from this is there were pesticides detected in every single orchard. They're focused mostly on fungicides, which are over sort of towards the right-hand side there. But there's actually a decent amount of insecticides that the bees are being exposed to as well. All right. What about risk? So this is the same y-axis that I was showing you before for assessment of risk. And now we're in a totally different ballgame. Okay? Now we're not below 1% hazard quotient, but we're upwards of nearly 80% hazard quotient for at least some of these orchards. And in fact, government agencies allow us to put a few benchmarks in here. So US Environmental Protection Agency says that 40% of an LD50 is an acute exposure level of concern, where all of a sudden now we've got to start talking to growers about this. European Food Safety Authority gives us the green benchmark, where that's the acute exposure level of concern, and then they also give us a 10-day chronic exposure level of concern in purple here. So they're actually saying, okay, if your bees are exposed to this, not just for a couple hours, but for 10 straight days, now we've got to, again, start talking to growers about this. Turns out 22 of our 30 orchards were above one of those levels of concern. So now we're in a fundamentally different conclusion compared to what we're just going out and looking at beekeeper colonies for. Now these bees that are going into apple are really exposed to lots of different pesticides. Okay? Yeah?
Yeah, I noticed your sampling sites are all near vineyards. And vineyards probably use more fungicides and pesticides and herbicides than anybody else in the state, but in the Finger Lakes region. So do you think your bees are picking up some of the fungicides from the vineyards?
It's a great question, and I'm going to get there in about 2 slides. So thank you for prompting me. Well, actually, we'll start to get there with the end of this slide. But yeah, yes, and I will show you how. So if you look at how the columns are shaded here, some are shaded light, and these were pesticides that were sprayed during bloom in apple, and some are shaded dark, and those are pesticides that were not sprayed during bloom, and that's the proportion of risk that came from each of those. The main take-home from this is 2/3 of the pesticides that the bees were bringing back were not actually sprayed during bloom, so they were not sprayed during the time that the bees were actually out there, which begs the question, where do they actually get these pesticides from? So if we start disentangling this question, so here are some relationships across all 30 different orchards. So here's percent apple area on the x-axis here, and just total pesticides that the bees brought back. So if there's more apple grown in an area, they are bringing back more pesticides. And also if they're bringing back more apple pollen, they're also bringing back more pesticides. Okay, so it seems like apple is at least part of the story here. But you have 2 different stories shaping up depending on what types of pesticides. So if we just look at fungicides, we do have this nice positive relationship where if they're bringing back more apple pollen, they're bringing back more fungicides. This makes a lot of sense because a lot of growers during bloom, they think that fungicides are not risky for bees whatsoever, so they're spraying during bloom for a fungal pathogen. Primarily apple scab is the main one, but a few other fungal pathogens that can infect the plant during bloom. But that's not the case if we look at insecticides. So insecticides, we have no relationship between the percent apple pollen and the number of insecticides or the hazard from the insecticides that are in the pollen. In fact, we have a positive relationship between pollen richness. So this is the number of non-apple pollens. So things like maybe grape pollen, a lot of different other types of wildflower pollens that the bees were collecting. So the more other types of pollens that they're collecting, the more risk they have. So the higher hazard quotient and the more insecticides they have in their pollen. Okay? So this is actually the project that Elena is working on right now. So we have this relationship. We think that the bees are potentially exposed to a lot of pesticides, not from foraging on apple, or say these insecticides, not from necessarily foraging on apple, but potentially foraging on wildflowers. What we're now what we've done this spring is we've gone out and we've said, all right, this drift of sprays that's occurring on apple, or potentially something in grape, or homeowner usage around in the area, potentially there's lots of different ways that some of the wildflowers might be getting insecticides into them, and it's those insecticides that are really toxic to bees. So what we've done is we're going out now and we're collecting, well, we've gone out and collected all of the wildflowers either within orchards, so you can see some dandelions blooming in an apple orchard right here, or different distances from those apple orchards to see what we actually find. What we're hoping that we can actually do here is we can say, okay, now we've got 30 different apple orchards. We've got growers doing lots of different things. They're spraying different things. They're spraying at different times. And they're spraying at different amounts. Can we actually relate that to what we're finding in the wildflowers, and can we make some recommendations to growers to potentially minimize risk to bees while still controlling their target pests? Okay. Happy to talk about this project further, either now or at lunch, but that's where we are with this particular project.
Why are bees more like exposed with wildflowers than apple flowers? Because the apple flowers has the same amount of pesticides, like it's the same pesticide that's been sprayed to apples than wildflowers. So why more exposure for wildflowers?
I think it's a really good question. So most of the pesticide risk, so if we go back to this particular risk, so showing the risk on the right-hand side figure here, most of that risk is due to neonicotinoids. So particularly thiamethoxam and clothianidin. Those are systemic insecticides, so they're very highly water soluble. So if they get into the soil, they can be taken up by the plant root and incorporated into tissue. That's wonderful if you're trying to target pests, because then the whole plant is essentially toxic to that pest. But it also can get into nectar and pollen. It's not like it just gets into the vegetative tissue. So I think likely what's occurring here, and we don't know, but likely what is occurring here is you have this drift for neonics especially that's occurring. It's getting into places that are not necessarily the tree. It's staying in the soil, neonics are also very environmentally persistent, and then because they're systemic, they can get into the pollen and nectar of those, pretty much any plant out there.
So it gets more concentrated in the soil than in the tree?
It depends. They're water soluble, so they can accumulate in particular areas. There's a lot of movement in the soil. They can dissipate, so they can leave the soil and actually be removed via groundwater as well. But yeah, it can be concentrated in some areas depending on the hydrology of that particular area. Yeah. Okay. All right. We'll move on from pesticides. Okay, so the second story, I'm telling you to think it's good for this. So the second story I'll tell you about, we have a few other projects working on pesticides. We're also working on management practices, habitat, and climate change, but maybe the most relevant for plant interested people is looking at some of our transmission work. So what I'll tell you about is a little bit about transmission of pathogens at flowers. This is some new research that we don't have published yet. Some stuff in the works, but not a paper on this yet. And then I'll tell you a little bit about why we think this is really important. So it's not just a fundamental question of trying to understand transmission, but how is this really important for pollinator health in general. Okay, so the main questions we'll go over are how do bees actually transmit diseases? What structures patterns of disease in plant pollinator networks? I'll tell you what that means. And then why is this important for pollinator health? Okay, so a postdoc in my lab, Pete Greystock, a couple years ago, or a few years ago now, published the first paper that really started to look into this and say, okay, bees have pathogens on them. Let's have our make-believe bee here with some pathogens in blue. So bees have pathogens on them, but we really don't know much about how they share those pathogens. There's lots of work showing that those pathogens are important for health, just like viruses and other pathogens are important for our health. But we really didn't know much about how they transmit those diseases. So Pete did some seminal work showing that when bees visit flowers, they sometimes slough off some of these pathogens. And then if another bee comes along, potentially of a different species, it can potentially pick up those pathogens and get sick. Okay? So floral transmission of pathogens is occurring out there quite a bit. So here's the bee picking up the pathogen. So a grad student in my lab got interested in this, Laura, and she went out and screened lots of different flowers, along with Pete, who started in my lab just last year, screened lots of different flowers for what bee pathogens were actually on there. So here's just a picture of a whole bunch of the different flowers that we screened. What we did is we screened for 3 different groups of bee pathogens, so microsporidians, 2 species of microsporidians, these Nosema species, a neogregarine, so one of these protozoan pathogens, and also a trypanosome, another protozoan pathogen. All 3 of these are known to be quite important for pollinator health and can also potentially be shared among plant species, among bee species. So our question was, okay, where are they on flowers, and how do flowers vary, or plant species vary in their abundance of having these pathogens on them? So here's some data showing prevalence of these bee parasites on different flowering species. So Daucus carota, so wild carrot, is our top one up there. If you look at proportion of bee parasites on those lots of different ones of those plants, it's somewhere up around 30% or so. Trypanosomes here, somewhere around, what is that, about 15% or so, and microsporidians the same, somewhere around 15%. So these are all different plant species here, and I've grouped them in ones that we have pretty good replication on, so 15 reps to 48 reps for spotted knapweed, which is the second one, and then when we have pretty low replication, it's down here. So there's variation across plant species for what species actually have bee parasites on there. And probably our biggest take-home message is that somewhere over a quarter of flowers, these are individual flowers that we have tested now, have bee pathogens on them. I think that's sort of a fundamentally new way of looking at flowers. These are not these beautiful things, well they are beautiful still, but they also are just riddled with diseases of bees. Over a quarter of individual flowers out there, go out into your garden and take a quarter of that garden and say, okay, that one has bee, all of those flowers have bee diseases on them. Pretty crazy way to start looking at flowers. Okay, so there's variation, there's lots of bee pathogens on there, at least bits of DNA from our screenings via PCR. Okay, so we went a little bit further with this, and we got a little bit more manipulative. So these are data from an experiment that Laura did last summer, and said, okay, are bees actually defecating on the flowers? So most of these parasites that bees share are fecal-oral transmitted parasites. So what she did is she took our common eastern bumblebee, Bombus impatiens, and then she inoculated it with Crithidia bombi, one of these trypanosome parasites. And what she found is that they do defecate quite a bit on flowers. So on average, she found feces on about 60% of the flowers in some of these cage trials where she let a bee interact with the plant and then go away. And you can actually see the feces. So we had fluorescent dye that we put into the bees so we could actually see where the bees defecated. And then this is important for the next slide. In general, it was about 10 microliters of feces that the bees were pooping onto these flowers. And then actually, interestingly, you can see here too, the defecation rate on the plants. If a bee was infected with one of these fecally orally transmitted pathogens, it did in fact significantly have more defecation. So you can think about that from the pathogen's perspective. It wants to be transmitted, so if it's a fecal-oral transmission route, it's a pretty smart pathogen for being able to induce the bee to poop a little bit more. Okay. So then what she did is she said, okay, the bees are pooping on the flowers. Great, that's a potential route of transmission. We've seen that varies across plant species. How long are the pathogens actually viable on flowers? So here, what she did is she took 10 microliters of feces, put it on the flower via just a pipetter, and then said, okay, how long is that Crithidia actually surviving on the flower? And the main take-home here, there is some variation, so it's viable for longer inside and then outside the flowers. It's viable on some flowers for longer than others. But the main take-home is just to look at the minutes. So it's over 3 hours that these parasites are viable on those flowers. And we know within a 3-hour window, there are going to be lots of other bees that visit that flower. okay? So there's, in that 3-hour window, there's definitely the potential for transmission, those bees actually picking up the parasites and taking them along with them. Okay, how about acquisition? So the bees have pooped on the flowers now, the pathogen has remained viable for about 3 hours. How about how well it is that the bees are actually acquiring pathogens from those flowers, so sort of closing this loop of transmission. So here again, she put 10 microliters of feces onto the flowers in the particular locations where she knew that the bees were defecating. And what she found is Clethidia cells started to grow inside uninfected bees quite a bit. So for all of these different species, so Lobelia, Lythrum, and Monarda, once you have those inoculations on there, the bees were very likely to pick them up, and there's also a little bit of variation across where the pathogen was on the flower, and also plant species, showing that some bees pick it up more than others, or sorry, they pick it up from flowers more than others, and also from different locations more than others. Okay? Okay, so hopefully that's convinced you that that pathogen transmission by bees, we've looked in as a field survey, and we've also conducted some manipulative experiments to show that this is likely occurring quite a bit out in the field. And now Bliss is following up with more stuff in the lab on this topic, which I'll get to in a minute. Okay, so what structures patterns of disease in plant pollinator networks? That's the next question. So we know that bees can transmit disease, but we have these hyper-diverse communities, so 416 species here in New York that are interacting with all sorts of plant species. Okay, so here's what a plant pollinator network will look like. If you guys have ever seen a network diagram, you can create gene expression networks. You can do lots of things with network diagrams. Ours just happens to be bees and plants. So here's Laura on the left and Pete and Paige who worked on this particular project. Okay, so what does this actually show? So here are 13 species of plants right here. So different flowers that are coming off of those different plants on the left-hand side. And then on the right-hand side are all the different species of bees that are interacting with those plants. So if you have a line that's drawn between any of those things, you know that that particular bee is interacting with those particular plants. So you can imagine this gets pretty complicated trying to figure out routes of transmission that might be occurring here. There's lots of different bees visiting lots of different plants. Superimposed on this in color are all of our different pathogens, okay? So this is the Nosema species, the microsporidians in blue, trypanosomes in purple, and neogregarines, the Apocystis species in yellow. And you can see that there's a lot of color. So what I've been showing you already is there's lots of pathogens out there. These are potentially being transmitted. Okay. So what Laura was interested in is saying, okay, we know there are things that structure that visitation network. One of them being what's in the landscape around. So you have to think like a bee if you're constructing these networks. And bees are not thinking about something the size of this room. They're thinking about something the size of a football field or bigger, right? So if you have different landscapes with different amounts of natural area to mostly agricultural area shown in those little circles up there, that might govern how that particular species of bee is interacting with different types of plants, okay? So she had 11 replicated wildflower plantings across this gradient of landscape simplification. Here are the networks from all of those different landscapes. So these are the plant pollinator networks without any pathogens superimposed onto them. You can see that they vary. So the ways that those bees were interacting with plants was varying due to landscape type. And then I'm not going to get into nitty-gritty details of all the results, but I'm just going to show you the main sort of punchline results from this analysis. We know that agricultural simplification decreases bee abundance, so there's more agriculture, there's fewer bees. That means that when you have fewer bees, that directly impacts pathogen prevalence. So you have actually greater pathogen prevalence when you have fewer bees there. They're interacting somehow and causing themselves to become more sick. But then you also have this indirect route. So when you have greater bee abundance, you alter the network, which negatively impacts pathogen prevalence, okay? So pathogen prevalence is driven in part from the landscape driving patterns of bee abundance, but it's also altering the way the bees are interacting with each other, which is then also influencing pathogen prevalence, okay? And I'm happy to, there's a lot of sort of under the hood interesting stuff going on in here that I'm happy to talk about, or we can not talk about it if you're not interested in it. All right. So, why is this important for pollinator health? I think this should be perfect timing. Okay. So let's go back to our managed honeybee. So it's sort of fascinating to think, I think, of lots of pathogens on lots of flowers, bees potentially transmitting disease. It's just a very fundamentally interesting question about how transmission occurs. But why is this actually important for actually trying to do something about pollinator health? So if we go back to our managed honeybee, this is our main, managed bee. There are literally hundreds and hundreds of millions of these bees here in New York. About 100,000 colonies in New York State. Let's say you have a beekeeper that isn't managing their bees well, and that colony is becoming, overridden with disease. On average, you have about 20,000 individual forager bees in a colony. And now if you have a sick colony, you have 20,000 bees that are going out into the wild and potentially interacting via flowers versus other bees. So yeah, what if beekeepers let their bees get sick? Here's varroa mite right here, which transmits deformed wing virus. You can see why it's called deformed wing virus over here. It makes the bee very sick. Okay, so now this is a different way of just showing a network. But here what I've highlighted, well actually here, let me just show you the network. So again, it's bees on the left-hand side here, and then flowers on the right-hand side. So each node is a particular bee species or a particular flower species. And what I've highlighted here is Apis mellifera. So this is now the western honeybee. So this is what beekeepers are keeping in their colonies. And let's say one of those bees got sick. So what it would potentially do is spread pathogens onto all of those particular flower species. What other bees are visiting those? Oh no. Ah, here we go, okay. So what other bees are visiting those? This is the bounce back that occurs. So in green now is highlighted the pathogens that are actually on those particular flower species, and then the bees that share pathogens with those honeybees that have visited those particular flower species, okay? So what we can start to do is essentially come up, let's see if this, there we go. So what we can do is we can start to come up with a prediction just based on visitation of who is actually going to be infected by what if you have a sort of a typhoid Mary that starts disease transmission down here. How is that disease going to spread? And clearly it's in part due to who is visiting what and that particular plant pollinator network structure. Okay? Okay. I think that's it. So yes, so one project talking about pesticides, one project talking about parasites and pathogens, and we can talk about a little bit about Bliss's role on that project as well if you're interested. Just to finish up very briefly, we have a website, so Cornell Pollinator Network. If you decide you guys are interested in pollinators and want to know more information about them, here's our URL. We also have a Facebook page, so if you're interested in becoming our friend on Facebook, you can join the Dyce Lab Facebook page. I think we have around 600 people who follow us right now. And then thank you very much for your attention, and thank you Bliss and Elena for joining me on this